Okay, so I will share my screen now. Okay. Okay, I hope you see. Okay, so hyperspectral imaging combined with machine learning to classify Flavescence dorée symptoms. So first, I try to be quick because I just noticed that I have just 10 minutes, but this presentation is longer, so I will be fast. Okay, so Flavescence dorée. Flavescence dorée is phytoplasma that affects, that affects vines, especially in Europe. It's transmitted by a grasshopper, which is Scaphoideus titanus, that feeds on the leaves and starting from spring. And so the symptoms develop during spring, but there are not leaves yet. So farmers actually recognize the symptoms when it's too late. So during August or July sometimes. The symptoms depend on the variety of the vine. So for red varieties, the leaves become red gradually, while for white varieties, the leaves become yellow. So it's more difficult to differentiate between Flavescence dorée symptoms compared to, for example, senescence. So what I'm trying to do right now is to create a system that is able to recognize leaves that already are affected by the disease, but the symptoms are not showing yet. Why is that? Because actually the state-of-the-art allows to perform chemical measurements on leaves. It's a destructive type of measurement conducted in a laboratory, but is effective only if the leaves is showing the symptoms already. So for asymptomatic sample is not effective. And this is too bad for farmers because it's not useful for them to recognize and to be sure of the presence of the disease when it's already too late. They want to know it before, and so they can plan the treatments and so on. So what I've tried to do is to recognize asymptomatic samples using hyperspectral imaging combined with vegetation indexes and machine learning. I will be quick on this. Vegetation indexes that I use in this work, focusing on red varieties, are these 10 vegetation indexes that are just quickly recapped here. mACI is an anthocyanin index that I've just modified compared to the state of the art. I've just modified it to span multiple bands, but it's the same as mACI, traditional mACI. They are divided between, for example, chlorophyll type of indexes, anthocyanin type of indexes, and so on. I checked the literature before choosing them because I'm not an expert on them, so I carefully read the literature about this. So what I did is, this is a measurement campaign that I conducted in 2023. I have new data for 2024 that I've yet to analyze. However, we acquired some leaves from this camp here, which is in Italy, near Pavia, which is in the Lombardy region. We conducted 3-time campaigns and collected some leaves from the camp. In total, we collected 100 leaves that we labeled according to the results of the chemometric test conducted in a laboratory. So to be sure of the asymptomatic samples, what we did was to check if in a later campaign, the plant was positive to the disease. So the leaves acquired in the campaign that were conducted before, if those samples were negative, then that plant, the sample was labeled asymptomatic because it wasn't showing the symptoms yet, but it showed them later. That's how we label the images. We use this camera that is pricey. It's called Nero's Hyperspectral ARVs. So it has a spectral range from 400 to 1,000 nanometers, and we tuned it to acquire an hypercube with a step of 5 nanometers. So at 120 levels for the hypercube. So each hypercube is actually a gigabyte and a half. So it's pretty much heavy on the disk. And the acquisition is not fast since this is lab equipment, basically. So it's from 30 seconds to 1 minute acquisition per hypercube. The acquisition setup was this one. We weren't able to acquire data in the field directly because the field was not powered by electricity. We tried it, but it was not possible. So what we did was collect the samples from the field with the aid of an agronomist, and then move to a nearby facility. It's like was like a little house nearby. So we mounted the equipment to scan the leaves. So we placed the camera to look at the floor, and we placed each leaf on a white panel to acquire the hypercube. We also acquired obviously the reference, which was a Teflon sample, and we normalized the white levels using a white background. So we didn't enclose this setup because we wanted to illuminate the sample using the natural diffuse light of the sun, to make it look as natural as possible. So we acquired the samples this way. In the next slide, I will refer to the spectra acquired from the hypercube as the spectra, but it was the normalized one that we acquired after normalization for the reference sample. Okay, so we have our hypercubes, but we wanted to exclude the background from them. So what we did was to recompile the color image from the hypercube and then apply segment anything model to extract the leaf from the background. So we basically applied this mask on the hypercube to remove the background. Then we extracted each pixel of the hypercube which belonged to the leaf and basically created this matrix here. I don't know if you can see my mouse. Anyway, this matrix here is like 120 columns for as many rows as the number of pixels in the leaf. So depends on the size of the leaf, obviously. Okay, so before proceeding, we wanted to check the sample variability. So I'm from a measurement lab, so this is actually the main topic. So what we did was to, before conducting every type of experiment that I will show you later, before anything, we collected 50 hypercubes of the same leaf sample, which was probably a healthy one, changing its position in the field of view of the camera each time. So 50 hypercubes, same sample, different position. We extracted the matrix that I showed you before, and we basically joined them together to create a bigger matrix. So by computing the mean and the standard deviation of each column, remember that the columns are the bands from 400 to 1,000 nanometers, we could compile the sample variability, which is this one. So in this graph, we can see the sample variability and the repeatability of the measurement together because of this, because of how we computed this mean and standard deviation. So obviously, the higher regions, the regions with higher variability are near infrared and green. Green because the leaf is green. Near infrared probably due to light scattering effects, because while we were moving the leaf, and the leaf is not completely flat, so the surface probably scattered a bit of the illumination. Then we can compare this overall repeatability with just one average spectra of among the 50, just to check if everything was done correctly. And the two spectra are actually overlapping, basically. So in the green spectra here, you can see the single acquisition, the average spectra of just one leaf, which includes only the sample variability, compared to the yellow one that also includes sample variability and repeatability of the measurement. To visualize this better, I've just picked some regions of interest and compared the two. So the values are basically the same. So what we can conclude is that the repeatability is very high for this type of measurement. And the differences between the two kind of curves are just due to the sample variability, not due to the measurement procedure. So we were happy to to find that, and we conducted more experiments afterwards. So just to recap what I said before, for each hypercube, I've acquired 100 samples, so 100 hypercubes. I've collected a matrix for each according to the dimension of the leaf. Then from these matrices, I computed just one. For each row of this new matrix, we have the average spectra of the leaf samples. So from the orange matrices, I've computed the mean, the average spectra, and I put it on the rows.
מצטרף, כן, אני לא חייב להיות בוובינר. דווקא שבאתי לשמוע מדבר שני ולא ראשון.
Okay. Then from this matrix, the blue one, I computed the vegetation indexes from that. So it was just a processing step to make it understandable everything, what I did later. So just 100 measurements, just 100 samples, not balanced, not enough to conduct machine learning. So what we came up with was a synthetic generation of the samples procedure based on a generative adversarial network. So what we did was using the original spectra, we feed them to the generative network to make it generate new samples. We add the noise. So the other noise was there to simulate a degraded measurement system. So basically, after training the GAN, we use it to generate 10,000 synthetic average spectra per class for a total of 30,000. And then we use those synthetic spectra to populate a new synthetic dataset, a new blue matrix, from which we extracted the vegetation indexes. And then we use this green matrix as the input for the machine learning model, which was an ensemble of 30 baguette trees. After training this model, we tested it on the original data. So we put the original data, the blue original matrix from which we extracted the vegetation indexes, and used that as the input for the test procedure. So the trained machine learning model was tested on those data, producing this classification confusion matrix. Here you can see the instances values inside the matrix and the other matrix on the left and on the bottom, on the right, sorry, and on the bottom. Remember that these are just 100 samples. So it's actually a very nice confusion matrix, but we also wanted to check if what we did was makes any sense. So we applied a Bayes test. I will not show you the math for that. Just trust me, trust the process. So basically what we came up with was using the results of the confusion matrix. We could say that the probability of predicting a sample as healthy when it is healthy was 98%, while at the same time predicting a disease sample as disease was 100%, so very nice. But for the asymptomatic samples that we want to classify them as disease because they are, still has some room for improvement because the probability of detecting as diseased an asymptomatic sample is just 67%. So there are already a few misclassifications that will be improved in the future. So this work is still in process. As I told you before, I collected new data in 2024 that I'm still elaborating right now because each hypercube requires a lot of work from my part. However, this work was a pivotal step for me actually to understand the phenomenon that I wanted to study. Also, I know that I created basically a closed loop, but it was just a test to check if this kind of procedure could work. So what I did in 2024 is conducting new campaigns, also on control data on different varieties. And I'm working now on the implementation of image analysis techniques to also retain the spatial information about the pattern of the disease, which could be useful to differentiate between symptoms of Flavescence dorée and other similar pathogens like Bois noir, for example. So I think I am finished, and if you have any questions, I'm open for discussion.